Hello, everyone. My name is Brennan Sullivan. I'm from the KDAM Epilepsy Research Lab here at Kennedy Krieger Institute in the Johns Hopkins School of Medicine Department of Neurology. I'd like to thank everyone for tuning into my talk. Today, I'll be presenting some unpublished data and talking about some ongoing experiments here in the KDAM lab. And the title of my talk today is An Early Forecast for the Syngap1 Storm. This is a large cohort study published by Dr. Ingrid Scheffer and the group in Australia, where they describe the median age at seizure onset for patients with Syngap1 mutations to be 2 years, with a range of 4 months to 7 years old. So with this cohort data, we know that seizures are occurring early in life in patients with Syngap1 mutations. So as a preclinical researcher, the question is, well, do mice with Syngap1 haploinsufficiency, do they also have early life seizures? And previously, all seizure data from this mouse has come from its adult age. So here I show in a P7 HETs, so these are haploinsufficient P7 mice. This is the mouse originated by Dr. Hugenaar, the original one, and the one that the KDAM group, we had previously published this year. These are early life seizures. They occur on the 7th day of life in these pups. I use a tethered subdermal scalp EEG recording for 2 hours. And here on those 1-minute traces on the left, you can see there's spikes, there's poly spikes and clustering of spikes, as well as seizures. Over here on the right, you can see a P7 wild type 1 hour trace, as well as a P7 het mouse trace. And this is a het mouse with a lot of seizures over 1 hour. When this is quantified for these P7 hets that were the same mice are recorded at P7 and then again at P12 for 2 hours each, you can see that they're ictal events per hour. I want to highlight that the females here are in pink and males are in blue. Currently, we have not seen a significant difference between males and females with their ictal events per hour. However, there does seem to be a group that at P7, they have a very high seizure burden. And there's another group of Syngap heps that have low seizure burden at P7 or even no seizures at P7. So with this being known that there are these seizures, and some of these seizures are short duration or other sub-seizure ictal-like activity, what we wanted to look at was there a behavioral phenotype that was different from the hets to the wild types? And so what we did was we just looked at a behavior on video with a score blind to genotype, age, sex, and seizure score of the hets, so blind to all those things, and they scored the behavior just on video of flexor spasms or jerks, which is denoted as 1, waddling, lordotic stretching is 2, or a behavioral seizure is 3. So when you look at this for the flexor spasms or jerks, there was a significant increase in flexor spasms or jerks in the het compared to the wild type. And then there was no differences between wild type and het for either of the other behaviors. However, when we took this, what we knew was scored but not by seeing the EEG or the true seizure burden, just the behavior, this behavior score. And we overlaid that to what we did know from the EEG of the true seizure burden. What we could see was that of all of the seizures, less than 50% had any behavioral score at all. So not only are these seizures subclinical, but they require EEG, as the majority of them were electrographic only. So this is P7 Syngap1 hets. So a study I would like to highlight, this is from Takao Hinch's group at Harvard in collaboration with Francis Jensen. What they showed here was using a chemoconvulsant-induced seizures with PTZ. It is a GABA-A antagonist. The idea is that the GABA-A antagonist decreases inhibition in the brain at high doses, and it induces seizures. So they induced seizures with a GABA-A antagonist. These seizures disrupted the plasticity of the auditory cortex during its critical period, where the auditory cortex undergoes refinement and the unsilencing of synapses in an experience-dependent manner. One of the things that they show that these early life seizures have a disproportionate impact on critical periods of plasticity, but also that AMPA receptor antagonists rescue these deficits in this acute seizure model. So with this being known, what I wanted to do was look at Syngap1 hets at P7 and see if I could stop seizures. So this is an EEG recording for 1 hour. These black lines are seizures. And you can see at 1 hour, I gave a loading dose of phenobarbital. Phenobarbital is a positive allosteric modulator of GABA receptors. It's the idea that it's going to increase inhibition. And when this occurred, it had little to no effect on seizures in hets, and maybe even aggravated seizures in the 2nd hour. I'm blind to genotype during my experiments, and so I wanted to show that there's even some, very few, but some knockouts live to P7, and I recorded from them. And when I do that after 1 hour of phenobarbital, when I gave it to them, they actually were aggravated by this loading dose of phenobarbital. So at the Kadam lab, we study these chloride cotransporters and early life seizures. So we've been interested in a chloride extruder, KCC2. And what KCC2 is known to do is extrude chloride, and this allows for the GABA-A receptor to be inhibitory. So because I gave phenobarbital, and there was no inhibition in the brain during EEGs, phenobarbital was almost aggravating these seizures. I then wanted to look at KCC2 to see if this was my suspect. And when I did some probing of total protein or its phosphorylation profiles, I did not see any data thus far that KCC2 is decreased in the cortex or hippocampus of P7 Syngap1 heterozygous mice. However, another way that I've been able to probe altered inhibition in the brain is, this is a paper that's currently under review from our group. It is a preprint. I used 2 phosphomutant mice. This is the S940A mouse and the T1007A mouse. They have mutations on KCC2 that don't allow for these sites to be phosphorylated, and they've been shown to increase and decrease KCC2 function, respectively. These mice were generated originally by Dr. Stephen Moss at Tufts University. So I gave this GABA-A receptor antagonist quite a low dose compared to what is normally used for chemoconvulsant-induced seizures. I gave 20 mg per kg, and what this did is this showed that in the S940A mutants, this KCC2 hypofunction mutant, that they went into status epilepticus. So I tried to repeat these experiments by giving PTZ 20 mg per kg to a Syngap1 het after 1 hour. And interestingly, the seizure rates went down. This was surprising. This was not what I was expecting. And what I think this may show or suggest is that early in life, there may be high GABAergic activity in Syngap1 pups. So after this PTZ experiments, I was a little confused, and so I went and started looking at the literature for what was known about PTZ and how its relationship to neurodevelopmental disorders. And so PTZ was used as a cough suppressant, and actually was used as an ECT therapy early on at high doses. So the cough suppressant was low. They gave high doses of the cough suppressant. They found it induced seizures, and they were giving this drug to induce seizures in patients instead of ECT back in the 1930s, I believe, and it lost FDA approval for its cough suppressant action, I believe in 1980. So this drug is not new by any means, and it is actually being used in low doses. So low doses are going to be given to children or adults and adolescents, 13 to 35 years, with Down syndrome. And the primary outcome of this study is safety and tolerability, but they're also using cognitive, functional, and behavioral paradigms to understand how this drug affects this patient population. And they're also using quantitative EEG and auditory event-related potentials to measure this drug's function. However, this clinical trial is based off of some preclinical work out of Stanford that looked at low dose, or identified that low dose PTZ rescued cognitive benefits in Down syndrome. in 2007. And then they also showed that using quantitative EEG, the Down syndrome mice had high gamma power on 24-hour video EEG, and that PTZ reduced this to wild type levels. And this is interesting because if you're familiar with our work, earlier this year we published in Biological Psychiatry that these mice, the Syngap1 mice on 24-hour EEG, also had high gamma. They had high gamma during sleep. You can see here, this is gamma power plotted. Yellow is wake, non-REM is blue. Gamma stays high during non-REM in adult Syngap1 mice. And then when we gave an AMPA receptor antagonist, this reduced gamma power and allowed gamma power to be modulated from wake to sleep back to wild type levels. So since this was, we initially showed this in the adult mice, we're now interested in younger ages. And so here is a P24 experiment that is a juvenile age of a wild type, which has this, again, this high gamma power during wake, low gamma power during sleep, and modulation over 24 hours on video EEG. This het mice has no modulation from, or very little modulation from wake to sleep, as you can see in this expanded transition from wake to sleep here. And then when we give this AMPA receptor antagonist at low doses, you can see we can rescue that to wild type levels. So this is at P24. So this is juvenile ages with a head cap experiment showing what we previously have described in the adult Syngap1 mice. However, since sleep structure changes over time and over age in mice, we ask the question, is this high gamma during sleep dependent upon sleep structure, or if we were to normalize the amount of time it was awake before the mouse was awake, before it went to sleep, would we get a robust response from this gamma? So using a telemetry system, we recorded during 24 hours, and we started off with 6 hours of sleep deprivation, and then the mice wild type or Syngap hets would then sleep. And what you can see here is that when we controlled for this duration of wake, that we got a robust response in this biomarker of gamma power, so 35 to 50 hertz on video EEG, and that the rate of change from wake to sleep for gamma was opposite in hets, and that the overall power is higher in hets for gamma, but importantly, the slow wave activity, or this delta power, 0.5 to 4 hertz, was not different. So even though we gave the sleep deprivation, the slow wave activity after the sleep deprivation was not different. However, the gamma power was. So in summary, these Syngap1 pups have early life seizures, and they are predominantly subclinical. And then some of the ongoing experiments have identified that maybe these Syngap pups have an increased GABAergic tone, and this actually may underlie some of these early life seizures. I've shown this through the phenobarbital aggravating seizures and low-dose PTZ actually decreasing seizures in Syngap1 hets. And then further, the recently reported biomarker of high gamma, so 35 to 50 hertz, which we had shown in our paper published this year, is present at juvenile ages at P24 in these Syngap1 mice. I would like to acknowledge from the Kadam lab, obviously Dr. Shilpa Kadam made all of this possible. Pavel Kipnis was a great lab mate, and Simon Emanuel, who's a lab alumni and collaborator who's at the Chang lab at UCSF, and also Dr. Richard Hugener's lab, specifically Dr. Iraqi and Dr. Hong, who allowed me to start my own experiments by generously giving us a portion of their Syngap1 colony. That obviously expedited my timeline to getting new results, and so thank you to them. Also, I would like to thank all Syngap1 stakeholders and the organizers of this virtual conference for young investigators for allowing me to speak, and I look forward to the discussion and questions in the future. So thank you all.